When norovirus enters the gastrointestinal tract, it utilizes the host microbiota to subvert barriers to entry and facilitate infection, as previously described in Learning Object 1. But this is only one component of the innate immune system. When norovirus infects an intestinal epithelial cell, alarms are triggered in the cell that cause it to secrete interferons and begin presenting viral antigens on MHC1 molecules. Macrophages and dendritic cells become activated when they phagocytize viral particles that bind their pattern recognition receptors, namely toll-like receptors, or when they become infected and viral components trigger cytoplasmic pattern recognition receptors. Antigen recognized extracellularly will be presented on MHC2, while intracellular recognition results in MHC1 attachment. Activated macrophages secrete interferons, which hinder viral spread. Tumor necrotic factor alpha, which initiates the inflammatory process, and cytokines, which call reinforcements like natural killer cells, neutrophils, and more macrophages. NK cells kill infected cells by releasing cytotoxic granules on their membrane. Reinforcement macrophages and neutrophils will continue to phagocytize what they can. Some activated dendritic cells will also secrete interferon, but unlike macrophages, dendritic cells will migrate to the nearest lymph node to present its found antigen to naive lymphocytes. This migration is the link between the innate and adaptive immune system. Naive lymphocytes enter the lymph node from the circulation, while the activated dendritic cell will enter through an afferent lymph vessel and travel to the T cell zone. This is where the naive T cells will pass by. If a T cell possesses the matching T cell receptor for the MHC antigen complex, it will become activated. MHC1 will trigger a CD8 positive T cell to differentiate into a cytotoxic lymphocyte and proliferate. MHC2 will trigger a CD4 positive T cell to proliferate and differentiate into Th2 cells. Very few Th1 cells will be produced during a viral response due to the cytokine profile. The effector cytotoxic lymphocyte will exit the lymph node through the efferent vessel and travel to the site of infection. The Th2 cells will progress further to the B cell zone. If a naive B cell from circulation has bound viral antigen and presented it on MHC2, then a Th2 cell will be able to bind the MHC complex with its T cell receptor. This is the secondary signal the B cell needs to become activated and begin clonal expansion. An activated B cell will differentiate into antibody-secreting plasma cells and a few memory B cells, which will be necessary to provide a faster immune response in the case of a secondary infection. These effector cells will enter the circulation. Some B cells will remain in the lymph node to undergo affinity maturation via somatic hypermutation, wherein B cell receptor affinity for the pathogen is optimized. Initially, IgM-class antibodies will be secreted by plasma cells, but since IgM has rather limited efficacy, especially during viral infections, B cells will isotype switch in order to produce more versatile classes like IgG and IgA. Back at the infection site, cytotoxic lymphocytes will kill infected cells since they are presenting MHC1. Evidence shows that norovirus has the capacity to infect not only enterocytes, but macrophages, dendritic cells, and B cells as well, so these cells will be killed too. IgG will exit the blood and encounter pathogens in the tissues. Here it can neutralize and opsonize free viral particles. Neutralization occurs when the antibody occupies all of the viral binding sites, such that the particle cannot infect a cell, and opsonization essentially makes it easier for macrophages, dendritic cells, and neutrophils to phagocytose the pathogen. Plasma cells can enter the lamina propria to secrete their antibodies. IgG antibodies will join the fight, but IgA antibodies will be shuttled to mucosal surfaces to protect against any norovirus particles in the intestinal tract. The delay between the innate and adaptive immune response is often days, and in that time, the host will have to deal with the gastroenteritis caused by the natural inflammation process. But in most cases, this is not fatal, and the subsequent adaptive response quickly clears the infection. Once the viral antigen is no longer present in the body, the effector immune cells will die off. Some memory B cells and long-term producer B cells will remain in the circulation to maintain high antibody levels, prepared to instantly leap into action should a second infection arise.